All right, everyone, let's dive into decoding brain resilience. Now, most people look at getting older and think their brain turning to mush is just a done deal, an immutable reality. But honestly, that's just not how it works. Aging is a complex process we can actually mess with in a good way. Take a look at the graphics on the screen. On the left, you see healthy myelin and active synapses. That's a brain firing on all cylinders. On the right, we've got frayed myelin, disconnected dendrites, and axonal degeneration. It's like the wiring in your house is starting to spark and short out. Today, we're going to look at the roadmap from normal aging to MCI, the epigenetic software of your brain, and the actual evidence-based stuff you can do to keep your head in the game. Here's the part that catches most people off guard: the latency period. The pathology, the actual damage, starts 10 to 20 years before you even notice a symptom. We've got this continuum. You start with normal aging. Maybe you're a bit slower at processing stuff, but your cumulative knowledge is still solid. Then you hit the critical transitional zone called mild cognitive impairment, or MCI. This is where you have focal memory or executive function issues, but you can still handle your daily life. If that keeps sliding, we're talking dementia. The numbers are wild. Global cases are projected to triple by 2050 to about 152 million people. The goal is to act while we're still in that gray latency box. So, what's actually driving the decay? Think of it as a four-pronged attack. First, you've got oxidative stress. Basically, your brain is rusting because it uses so much oxygen, which creates ROS that damages your DNA. Second, neuroinflammation. Your microglia, the brain's immune cells, get chronically activated and start pumping out toxic cytokines like IL-6. Third, mitochondrial dysfunction. Your cells' power plants lose efficiency, leading to a massive energy deficit. Finally, vascular compromise. Your blood-brain barrier, the BBB, gets leaky and blood flow drops, so nutrients can't get in and waste can't get out. It's a mess, but understanding it is how we fix it. Think of your DNA as the hardware, but epigenetics is the software. It explains how your lifestyle can actually turn genes on or off without changing the hardware itself. We've got three big players here. DNA methylation, where adding methyl groups silences neuroprotective genes like BDNF. Then there's histone modification. HDACs remove acetyl groups and basically zip up your DNA so genes can't be read. Finally, non-coding RN adds silence messages before they can even make proteins. The good news? We can target these switches to keep your brain's plasticity alive. Let's talk tools. Omega-3s are your brain's structural architects. Your neuronal cell membrane is a phospholipid bilayer. If it's full of saturated fat, it gets rigid and signaling slows down. But when you integrate DHA, it creates kinked tails that keep the membrane fluid so signals can zip through. Then, you got EPA, which competes with pro-inflammatory acids to keep cytokines in check. Meta-analyses show these have positive effects on non-demented adults and MCI patients. A typical dose is about 1 gram a day of combined EPA and DHA. Curcumin is a total beast when it comes to being an epigenetic modulator. It can inhibit DNMTs and HDACs, basically keeping your chromatin open so your neuroprotective genes can stay active. It even boosts BDNF, which is like miracle grow for your neurons. But here's the reality check. Native curcumin, the stuff you just find in a spice jar, has terrible bioavailability. Your liver just kicks it out. You have to use a formulation with piperine or a liposomal delivery system to actually get it into your circulation and across that blood-brain barrier. Without the fix, it's just expensive yellow pee. Next up, the vascular guardians. Resveratrol and polyphenols from things like berries. These work by activating SIRT1, the famous longevity gene, which regulates your mitochondria and helps your cells handle stress. They also improve neurovascular coupling. That's just a fancy way of saying they make sure your brain gets enough blood flow and oxygen exactly when it's working hard. Plus, they help calm down the inflammation pathways like NF-κB. Studies actually show improved cognitive performance in older adults who take these consistently. All right, let's get into some fungi. Lion's mane. This is the mushroom frontier for brain health. It contains compounds called hericinones and erinacines. Its big claim to fame? It stimulates the synthesis of nerve growth factor, or NGF. NGF is essential for maintaining those cholinergic neurons that keep your brain sharp. It even promotes the repair of myelin sheaths and helps expand your neural networks, literally growing more connections. Pilot studies on MCI patients have already shown improvements in cognitive function scores. B vitamins are what I call the homocysteine regulators. High levels of homocysteine are basically neurotoxic. They lead to brain atrophy and vascular damage. To fix this, you need a trio: vitamin B12, B6, and folate, working in synergy. They move homocysteine through the remethylation and transsulfuration cycles. The VitaCog trial found that lowering homocysteine significantly slows the rate of brain atrophy. If your levels are high, this protocol is absolute synaptic gold. Look at this pyramid. Supplementation is the tiny tip at the top, but the base, the part that's non-negotiable, is your lifestyle. We're talking about dietary patterns like the Mind diet, which is a hybrid of Mediterranean and DASH. It's all about leafy greens, berries, and fish to kill systemic inflammation. Physical activity is also huge. It increases BDNF way more than most supplements ever will. And don't forget cognitive reserve. The use it or lose it principle is real. Keeping your brain busy with complex mental tasks acts as a buffer against damage. Time for some straight talk. Navigating the noise. A lot of the hype you hear comes from in vitro studies. That's just stuff happening in a petri dish. Those studies often use concentrations of supplements that you could only get via an injection, not by eating them. Also, most clinical trials only last 12 to 24 weeks. Since cognitive decline takes decades to happen, it's hard to prove a 3-month trial stops dementia. Plus, a lot of these studies have small sample sizes. So, while the science is exciting, we've got to be smart about the bioavailability gap and the quality of the evidence. If we're building a protocol, it's got to be multimodal. There is no silver bullet. For structural integrity, you want omega-3s. For epigenetics and inflammation, get bioavailable curcumin. For vascular health, stick with resveratrol and polyphenols. If you want to boost nerve growth, lion's mane is your go-to. And for metabolic health and homocysteine, you need that B complex. You're attacking the problem from every angle, which is the only way to really win. Last takeaway, the window of opportunity. Look at the timeline. If you wait until age 70, when symptoms appear to get a diagnosis, the pathology has already been cooking for 25 years. Prevention isn't about stopping aging. Nobody can do that. It's about building resilience. Your brain's fate isn't just written in your DNA. It's edited by your daily choices, the food you eat, the way you move, and the supplements you pick. Don't wait for the silent phase to end. Start editing your story today. For those of you who want to nerd out on the data, I've included the selected references on the screen, everything from Frontiers in Aging Neuroscience to JAMA and the Cochrane database. The science is there, the pathways are mapped, and the tools are in your hands. Thanks for listening, and let's keep those brains sharp.